Okay, so today will be a little bit of a ride, and I'm just going to apologize up front. You're not supposed to do this as a speaker, by the way, but I am going to apologize up front and say that we're going to finally delve into some science as we go through this. And the first part of this will be more general in nature, and it will become a little bit more scientific as we go, but I will explain the concepts at the top level and try to explain why the position, the young earth position, is the way it is. So I wanted to start off, as we've talked about the biblical discussion, the theological interpretation of Genesis 1, and Genesis 1 through 11, Exodus 20:11, Psalm 33, and everywhere else in Scripture that refers back to the doctrine of creation. It's not about a special genre. And we ended last week by discussing why we think the day position is the strongest position in Scripture. That a day is 24 hours. That is the most straightforward and logical position, and hermeneutically faithful in interpretation of Scripture. So we talked about that, and we talked about the other views as well. However, we also understand that in the PCA, we have four different accepted views on that position. And one of these I'm going to talk about today is not related, it's related to every one of them, including the 24-hour creation perspective. And that is the intelligent design movement. Intelligent design does not mean young earth creation or old earth creation. It is a position about an observation of the design of the universe. And so I'm going to explain a little bit behind it, a little bit more detail. I think most everyone in here is inherently holds to the position of intelligent design. It is consistent with the biblical position. So I want to describe it and also talk about some of its advocates and the perspective here. So it's important that when we think about evolution and the evolutionary perspective, when we look out as scientists, as we look out at the world around us, it looks designed. This is the this is the natural bent we have when we observe it. In fact, it's so strong, this natural design, that some of your leading scientists, evolutionists, have to remind the evolutionists that it is not designed. So a great example here is Francis Crick. Francis Crick, who some of you are aware of Francis Crick, who is the Nobel laureate who co-discovered with Watson the structure of DNA. And Francis Crick said biologists must constantly keep in mind that what they see is not designed but evolved. So this is the trick, right? This is the inside scoop. You have to bias yourself up front that no, it's not designed. So what is intelligent design? So I'm going to talk a little bit about the history, key leaders in the organization, the key concepts as well. Now, so what is it? ID is short for intelligent design, holds that certain features of the universe and of living things are best explained by an intelligent cause, not an undirected process such as natural selection or random mutation. So I think we're all on the same page here. This would be very consistent with the biblical perspective of an intelligent cause. Now, you can be a Muslim and hold to that position. You can be anything but anything that holds a perspective that there was a God who created the universe. You can even be the ancient animistic pagans and say, well, it was intelligently designed. So what this is really different from is the modern view that evolution, the natural evolution, the materialistic perspective that denies any intelligent cause beyond the physical universe. So this you can go back to 1985, a British Australian biochemist. He wasn't a young Earth creationist at all, but he started recognizing some of the problems associated with evolution. So in the 1960s, what happened in academia, you had the biochemical revolution. So evolution had been developed in the universities and understanding phylogeny and cladistics and so forth in the universities. And in the 1960s, after the discovery of DNA with Watson and Crick, and understanding what's going on at the biochemical level opened up all new understandings of what biology was all about. And at that point, when we started understanding what's going on inside the cell, rectifying that and correlating that with what the larger biology and organismal sciences at the larger level and the phylogeny, I can't even say it now. I just said it. Phylogeny. And the cladistics and all that was developing at that time. And now they had to rectify those and bring those together. And that was a problem. Trying to understand how this was actually going to occur. And simultaneously, about the same time, you had the famous Miller experiments where they were doing test tube, life in a test tube, where they were putting electricity through the test tube and seeing if they could make any molecules of life and some of that. And I will come back to that. So all of this was happening in the 60s, and it culminates in a few folks started noticing the problems. And some evolutionists, like Stephen Jay Gould at Harvard, would say the problem was so severe that he adopted a new position in evolution called saltation. And if you're familiar with that idea, it is where there's a constancy in the species and there's some traumatic event that makes them jump to the next level because we don't observe the transitionary fossils. So here's your lead evolutionist and lead paleontologist in the world at Harvard leading the charge and say, well, this is why we don't see transition fossils. We can talk about Archaeopteryx, and it's a separate thing altogether, and that is sometimes at the lower level, and I say that at the lower level in the popular culture held to as a transition fossil. However, it seems to be complete in and of itself, and just like the duck-billed platypus, it doesn't make sense from an evolutionary perspective, but from a design perspective, it makes sense from a creator who wants to create a diversity of different animals, but you don't. You can always arrange life. I can find, so here's an aside, I can find a bunch of horses that look, or equine species in the fossil record. And I can see very small ones, and I can see really big ones. And then I can arrange them from small to big. I say, oh, look at that. You know, or, and you can say it's diverse like that. Or I can think of it's like a dog, and I have big dogs and small dogs, and they all come from an ancestral dog, and some became small and some became big, depends on the interbreeding. So it's all how you arrange it, and the bias you bring to it, and say, oh, look at the diversity, and I'm going to arrange it from small to large, and look, this is how evolution occurs. So that was prior to the cladistics and the biology, that was the just so story of evolution. It was arranged that way. Yes, Bob?
Yeah, just a quick point. I seem to recall they call that quantum evolution. The idea being that a reptile laid an egg and a bird popped out.
Yeah,
the paucity of evidence was so bad that they had to go to a quantum idea.
Yeah, so, right, and this was not quite the that perspective. That was Goldstein's Hopeful Monsters. That was the idea at that time, and as not seeing the fossil records, and later became known as quantum evolution as a nickname. However, what saltation is, because you have millions of years, instead of having this gigantic leap in one generation, now that they're saying there's conditions, say a magnetic reversal of the Earth's magnetic field, that during that period of time, over a series of a few, maybe 1,000, 100,000 years, is when that saltation occurs. But on the grand scale of things, for millions and billions of years of the Earth's time frame, you would not see that in the fossil record because it would be such a short period of time, and so therefore you don't see transition fossils. So it became an argument from the absence of evidence, so which is problematic. But getting back to where we are here with Michael Denton, recognized some of these problems and started pointing out even the phylology. I'm not going to even say it anymore. And we understand that this is looking at when you look at the comparative structures of animals, right? And you compare their structures and say the relationship between them. And this is the physical characteristics that we're getting at. So he published this, and he said a theory in crisis. Now, evolution was not a theory in crisis from a social perspective. From a social perspective, it was entrenched in universities. It wasn't going anywhere. There was no paradigm shift in the thinking in the universities or so forth. This was the beginning of those scientists recognizing the fundamental problems of evolution in the modern period of time after the biochemical revolution. Charles Thaxton, who's a PhD in physical chemistry at Iowa State, postdoc at Harvard and Brandeis, he has also had a problem with this, and you'll probably hear his name as well, Mystery of Life's Origin in 1984, and he was also the editor of Panda and People. So when did this really break into the mainstream? When did people really start understand what was going on? And that was Philip Johnston. Philip Johnston published this. This was a runaway bestseller, New York Times, and it was Darwin on Trial. And he had his BA in English from Harvard, and his Doctor of Jurisprudence from the University of Chicago. So he was a law professor, and he was looking at the evidence from a perspective of law. Okay, let's bring the evidence together. I've done forensic trials. Bring the evidence together and bring it forward, and let's actually review that. And so he's considered the father of the intelligent design movement. He brought together a number of people and professors from across top universities around the country. They all met out in California at the time and had this conference of top scientists at the time, not creationists. This is coming out of the academic world, and it was an internal revolt within their academic world to evolution. Some of them were not Christians, and some of them were, but it was that perspective. Dean Kenyon, Professor Emeritus of Biology at San Francisco State, and he had his Bachelor's of Science in Physics from the University of Chicago, and a Biophysics from Stanford, postdoc at UC Berkeley. No small potatoes here, by the way, folks. So this is not a bunch of kooks that went to sometimes brandished as creationists going to these very kook schools. You know, that's the name the pejorative way in which the evolutionists like to portray creationist, intelligent design folks. They're ignorant, and this is not the case. He co-authored Biochemical Predestination of Pandas and People. He is a young Earth creationist. So you have a mixture. Intelligent design does not mean you're young Earth, old Earth, or anything like this. It means that you understand the world that was created by an intelligent designer. And then finally, in the scientific side, we had Michael Behe in Darwin's Black Box. This has had several iterations of this book, and this is not the current copy. That was the original when it came out. Darwin's Black Box. He had his PhD from the University of Pennsylvania. He introduced the idea from a scientific perspective and the biochemical perspective of what is known as irreducible complexity. In other words, a system at the biochemical level is so complex there is no way, no steps, sequential steps to get there. You had to bring all the pieces to get there all at once. So there's no evolutionary way to assemble a specific protein, for instance, at the biochemical level. There's just no way to do that sequentially. You can't do it one amino acid at a time, and I'll explain that. And he was the professor at Lehigh University, testified in several court cases. There's others. Stephen Meyer, you may be familiar with, their books, William Dembski, Information Theory, Paul Nelson, Jonathan Wells. All of these are well known. The Discovery Institute, if you hear anything from the Discovery Institute, that is the institute about intelligent design. And the list goes on. Some famous politicians, Michelle Bachmann, some famous scientists, and some I've already talked about here, Sam Brownback, Pat Buchanan, Kirk Cameron, Charles Colson, Ann Coulter. All of these folks have said that they believe in intelligent design and advocate for it. Right? So James Dobson, the late James Dobson, Bill Frist, Chuck Grassley, John Hagee, Mike Huckabee. Of course, Joel Osteen's on here, so I hope that doesn't ruin it for you. But this is the list of folks. Chuck Norris. So all sorts of. I'd hate to be in a debate with Chuck Norris on this. But these are advocates of intelligent design that are out there. So what are the key concepts? The origin of new function and parts of living systems is what we're getting at here. The origin of life itself, also known as abiogenesis. So we can talk about it from a cosmic perspective, we can talk about it from a biological perspective, but for the most part, the intelligent design movement focuses on the biology of things, and that's where it had its, really its beginning was in abiogenesis. And abiogenesis is the genesis of life from non-life, or spontaneous generation. You might have heard it mentioned that way. And it really is a discussion in that area. And it also talks about the origin of information. When we look at the DNA, it is a blueprint, it is a code of how life is supposed to work. It's like reading a book. There's words and sentences, and you put them together and you make different proteins. And so there's letters and words and sentences, and even up into complete concepts, if you will, in life, in the development of the DNA, so that you make very complex organismal systems because of how you tell the system to behave. And it's information encoded on the DNA. So natural selection. So this is important as we look at this, we keep this in mind as well. One of the key driving forces of biological evolution is this idea, this concept that you have natural selection is going to drive, you have mutation and natural selection. You have a mutation, and if it's favorable, natural selection will then favor that mutation. So that's the evolutionary concept of natural selection. And they also have this other concept of genetic drift of an entire population. If you look at the genetics of that entire population, it drifts over time. And so if you have a dog family that moves to Siberia, not only will will it favor those things that grow long hair in that type of environment, and those that have short hair will not be favored in that environment because it's much colder. So you'll have that genetic drift driven by the environment that's inherent within you can have a short-haired dog and a long-haired dog from, a pair. And just when you look at the diversity there over time and the entire population, that's going to shift if it's in the northern environment toward more long-haired dogs. And so that's the idea of genetic drift. But there was no mutation in that. Now, if there is a mutation that actually gives a favorable benefit, and now that this one dog has extra fat layers, and because they're in that environment, because of this mutation. Now, one, it's very difficult to have one mutation that causes this type of thing. This is part of the problem with evolution. But let's say you did, and let's give that. Then that would be favored in that type of environment. Usually, those are deleterious. They cause problems. And in a southern environment, those dogs would die off because they probably have heart disease faster or something along those lines. So, but this is the idea of natural selection. And so gradual over time changes, and these changes must produce a survival advantage and be selected and passed on. The process by which species adapt to their environment, I've talked about this. Difference in survival between different genotypes is the reproductive success. So the evolutionists talk about this reproductive success as they move forward. We're talking about natural selection. So understanding this is important in this conversation. Charles Darwin says, if it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down. He says, if it could be demonstrated. He says this. And so what we'll look at then is what Michael Behe really talked about in Darwin's Black Box, which was just a just so story. It was nice to say, well, this evolved into this and the. Well, how do you know? Well, because it looks like it. We arranged it in a specific order. It says, okay, well, let's look at it from a biochemical perspective. Biochemical and molecular revolution since about 1960 has changed the way we understand this. How does natural selection work at the molecular level? Certain complex functions appear to be irreducibly complex. This gets to the example that Behe uses is the flagellum. There's actually a number of different ones he uses. This is the flagellum of a bacteria. And to understand the complexity of how this flagellum works alone by itself, it has 40 different protein parts. A stator, a shaft, a propeller, the proton motive force, and the gears, all of that goes into this flagellum just to make it turn. It's got to have the energy to turn and spin. It has to have the bushings. It has to have all the stators, everything you would think of, an engineer would think of to actually spin a propeller is the same thing. It has to go in here. And all of those proteins, every single one of them are complex even of themselves. This spins at 100,000 RPM. So from an engineer perspective, think about what's going on here. It has to be able to stop in a quarter of a turn. In a quarter of a turn going at 100,000 miles RPM. There is no full Darwin explanation on this. There's no understanding of this. There is no stepwise approach to get to this. And 40 protein parts, each one of them having hundreds and thousands of amino acids to make them up. And so you can't say, well, we first evolved the stator, and somehow evolution says, well, the stator, we're going to select it because it has a purpose. There's no natural selection for just the stator by itself. It would be a waste of biological energy just to make a stator for some intended purpose that would come up in the future. It doesn't even know where it's going. Evolution is blind. It doesn't have a purpose in an end state. It just is naturally selected. It's just genetically driven. So you just produce one stator here by itself. Why? What would be the purpose of just having the stator protein in this? You would actually have to evolve all of them at the same time to have any function to be selected for in the first place. That's the irreducible complexity problem. Now, the counter view, just wants you to know what evolutionists have tried. They says, well, maybe it was the needle nose pump, and it has only 10 of the 40 parts. It doesn't help solve the problem. It just shows some similarities of function across different biological parts. You have some proteins there, you're 30 proteins away, and hundreds of thousands of mutations away from getting to the flagellum.
I ain't gonna reach the 30.
Yeah, exactly. That's the problem. Still needs all the precise parts and the precise assembly. So I didn't even talk about the assembly of putting all that together.
Right. Yeah.
So that's the, having it all functioning is one thing. Think of the entire factory and the machine behind it to assemble it just right, at the right place, at the right time, and all the proteins for that. This is truly amazing when we see God's handiwork at the molecular level. It's an amazing story. Then the problem of origin of information. No natural cause for the appearance of information. Consider SETI is a good example. We're looking for, we have all this money invested in looking for UFOs that are out there, right? We think they're out there. I don't think they're out there. Some people think they're out there. And we have all these programs designed and turned the antennas toward deep space, looking for any type of information. False alarm after false alarm after false alarm after false alarm in the SETI program. Oh, we think we have information. No, it's not. It's random. It's random. It's a pulsar. It's a two suns rotating around each other, and then there was a planet involved, so we thought it was. Anyway, all these things happen over and over again in the SETI program, and we've yet to find any information in the SETI program, but we're looking for specific arrangement of information from deep space. That's what we're
Hollywood tries to do a big deal.
Yeah, Hollywood tries to do a big deal of it. So there's a design, just so what you understand, design inference. What do we mean by the origin of information, whether that's in the genetic code or whether that's in deep space? It's an improbable object or event. It has to have extremely low probability. But that's not just enough. The probability in this room that any one molecule of oxygen that I breathe in, out of all the oxygen in this room, is very, very low. But that's not enough. I can still breathe it in, and it'd be even though it's a low probability event. So it's not just about low probability. There has to be specification with it. Recognizable pattern. So you have this combination of low probability and specification that actually gives you information. And we inherently understand this when we look at Mount Rushmore and we say, that didn't just happen by erosion there on that mountain. There was a designer that got up there and did something with that. Now I can see, and we can go out and we name a rock, old man's rock, because it looks like an old man, because it weathered just right and stuff. That's a little different than when I go to see Mount Rushmore, and it has the faces of 4 different presidents.
That rock is no more. Boy Scouts knocked it over.
Yeah, right. And you hear this often, the works of the sonnets of William Shakespeare. What's the chances that a monkey could type one of these randomly on an actual typewriter? So you have to have low probability and specification to get the information, and inference is the best explanation. Inference to a intelligent designer doesn't say who the designer is. It says it has to be a designer for this, or inference. Somebody had to make this information. So I'm going to jump now into the biochemistry aspect. I'm going to be on my home turf a little bit here, and so I'm going to try to explain some of this because there's some important distinctions here that you can understand. So biochemistry is the study of chemical processes in living things, the very basis of life. Since we're talking about abiogenesis, this is important. The structure and function of small and large molecules that makes up the cells. So when we talk about structure and function, biochemistry is structure and function. We look at the relationship between the two. So the DNA double helix is an example of that. You see it all over. You will use it everywhere in pop culture now, the structure of the double helix to symbolize life. Protein folding and its associated structure membrane organization on the cell membrane. Some of you might remember that from your biology class. Membrane organization, it's adhesion to each other, cells forming with each other. Then in function we get into, well, how does it work? The catalysis, or when we talk about catalysis, we're talking about the actual making a reaction speed up or slow down. These type of things. The catalysis of the chemical reactions of life. This is photosynthesis, and it's all of the metabolism in our body. It's the cellular transport, the protein synthesis, immunity, gene expression. All of these things fall into the function side, so it's relating structure and function. Bob's is enjoying this very much. This is like going back to his back to work. All right. So my fellow biochemists in the room here. So of course, there are classes of biological molecules. Carbohydrates, and your carbohydrates, low carb diets, you know, you're familiar with that in pop culture. This is your sugars, your polysaccharides, your starch. But you also have carbohydrates in DNA and RNA. This is an important. DNA and RNA are complex molecules that have several components, of which carbohydrate is a part of that. I'm going to come back to that. This is very important. Lipids. Lipids are your fats, right? They compose the membranes, adipose tissue, and so forth, and it's your cellular energy storehouse in your cells. And then nucleic acids, your DNA and RNA. That is the information. Everything is coded in that information. So when we get to abiogenesis, we're saying here, spontaneous generation, as I mentioned, that this is the development of early and simple life from inorganic chemicals. Inorganic chemicals. This is just your carbon dioxide, your minerals in the ground. That's your inorganic chemicals. So somehow out of the ground, out of the earth, came life. So how does that happen? By way of random and natural processes, without the interventions of contrived forces or design. That is abiogenesis. That is the predominant view in the universities today. It is organic. What do I mean by that? Chemical compounds containing carbon, and inorganic is everything that doesn't contain carbon. Our bodies are made of carbon. So how do we, as humans, come out of the ground that's predominantly not carbon? And we'll look at that for a second. And not only that, we have this concept of entropy. Now, some of you, especially parents, are used to seeing entropy happen on a regular basis in the kids' room. Entropy is that spontaneous direction towards disorder. Entropy is that idea of breakdown or disorder that appears, and it's a natural. It's the natural process. It's one of the laws of thermodynamics. Everything proceeds in this direction, and so everything is running down. You've heard people talk about the heat death of the universe in some cases. Everything is running down because of this law of thermodynamics known as entropy. And it's a natural process. And to fight it, to go the opposite way, you have to input energy to make it organized again. So you got to tell somebody to go clean up the room, you know, and they have to expend energy to make it organized in a structure again. Yes.
Directed energy.
Yes, directed energy. That's right. So, and that's a good point that I'm going to come back to. So it's very specific. And of course, if I just applied random energy to that, it would just get worse. In fact, if I applied random energy to that, it'd probably burn up, right? Catch fire. And then it would just all be ashes. So, and in some cases, that might be good. But in this case,
Then you would have to arrange it.
you might have to. But this is the idea of entropy. It's the natural process of things. So how do you get from, it's interesting. So the Earth's crust is in blue. If you just look at the different elements on the chart, you say, well, what is the component of the Earth's of the Earth itself? If life came from non-life, well, let's look at that. Let's look at all the main components of life and what those elements are, and see what the arrangements are in of the earth. Well, here's your elements. Most of it, you have a lot of oxygen, right? And a lot of calcium and sodium, and a lot of silicon, and others, all your other inorganic elements. And life is pretty much carbon, oxygen, and hydrogen. This is your relative percentage relationship between them. So how do you go from something that's arranged like this, in the blue, to something that has a percentage that's in the red? Randomly, without any direction or any process, right? This is the challenge for abiogenesis. The other concept we have to think about too, this is really getting into some chemistry here, is what we call an activation barrier. Every time two molecules want to come together to create another molecule in a chemical reaction, because this abiogenesis is about chemical reactions, so every single chemical reaction has what is known as an activation barrier. In other words, there has to be a certain amount of energy that's provided to the system to overcome that barrier to get to the product. This is going to be a higher energy, this is going to be lower energy, and it wants to go downhill. Entropy again, right? So it's going to stay up here as a stable product, but over time, it'll warm up enough or absorb some type of energy from the sun or something like that, that overcomes that barrier. Think of this as a bowling ball on a hill, and it's in a little gully. And then something comes and knocks it just enough to get over the hill, and it's going to go down to the lowest energy state. So everything's going to run down. This is why entropy works this way. All chemical reactions work this way. It goes down to the lowest energy state. Now, you can imagine if you're going to try to go the other way, you're going to have to push that bowling ball all the way up the hill. So if you want to go from a low order to a high order system, you have to push much more energy, very directed energy, to get it uphill. That's the basics of most chemical reactions. You have some, and the difference between what's higher and lower is different for every chemical reaction.
You spend more energy going uphill than you would.
Yes, and going downhill is entropy. Going uphill is directed energy to get it up to a higher level. So that's, now, this, I didn't put any of the chemicals in there. This is your standard biochemical pathways just for metabolism in the human body. So you have the Krebs cycle, you have glycolysis, you have carbohydrate metabolism, you have protein synthesis, all of that, and this is just one small snippet of that pathway. And what's not on here is things like photosynthesis and so forth. And every single one of these dots represents a chemical in the body for metabolism, and between the dots is a connection, is a chemical reaction that occurs that is catalyzed by a protein. A very specific protein for every single one of these chemical compounds in the body. And this is just the metabolism that's going on. Not the signaling pathways, not any of those other things. This is just the metabolism that's going on in biochemistry. And each one of those has a protein. These proteins are very, very, very large molecules to do this. Just like the flagellum, some are larger than the flagellum that you already saw. And some of them are like ATP synthase. Just to make the ATP that drives the energy of the body is larger than you saw in the flagellum. So these are very, very large chemicals. So of course, when I taught biochemistry, my students had to, just as an aside, their final for the biochemistry class was a paper, a blank poster paper, this that looked like this, and it was empty, and they had to fill it in. That was their final.
Yeah.
I put a few arrows in there, and I gave them a few, anchor points, but that was it. That was their final. So they saved it too. When they got it back, I still have my test.
How was your curve?
I did curve it.
Yeah.
I told them at the beginning of the class
That's what
that this was going to be their test, so they knew the whole class that this was coming. And then we went through every part of it. Yes, Bob?
You bring back memories. On my very first day of Biology 101, 54 years ago, the professor drew the Krebs cycle on the blackboard, and he said, there'll be a test on this tomorrow. To do exactly what he said. I almost said, I'm going to become a philosophy major after that.
Exactly. All right, so where am I going with this? So this is a part. I just want you to understand the complexity, the amazing complexity of what's going on in the cell, and why evolutionists are having problems with this, but they deny the issues. Now, so which came first here? This is a problem. So when they think of the standard answer for life is you have, how do you make DNA? How are you going to make DNA? Well, I need proteins to make DNA. Well, how do I make proteins? I need the DNA to code for the protein. So when you have the whole issue of what came first, if I need DNA to make life, how does it make life if I need the protein to make the DNA and I need the DNA to make the protein? And it goes through the RNA step is the translation on that. So this is the proteins are necessary to replicate DNA. Proteins in DNA are necessary to make RNA. DNA in other proteins are necessary to make proteins. So how do you evolve DNA which makes proteins if you need proteins to make them in the first place?
Right.
And how do you evolve proteins if you need DNA to make them? This is the conundrum. So this brought up the life in the test tube, this whole idea. And so just so you realize, life was not made in a test tube. What was made in a test tube was things like methane. So there's a carbon molecule in here that wasn't in the normal atmosphere. Actually, they're using methane, a reducing atmosphere, and they come up with things like formaldehyde. So they run electricity through this, and they come up with things like formaldehyde. Of course, that'll kill you, but yeah, preserve you fairly well. But they'll say, well, that's an organic molecule. We're able to make organic molecules if I put an electrical discharge through a reducing atmosphere, because the whole Earth was supposed to be in this reducing form before there was oxygen, right? Life came about in a reducing atmosphere, And so they use this reduced atmosphere because they feel that it's more conducive to making life, but then they run into other problems. And so then they do this, and all they've come up with is very simple organic molecules. That was the Miller experiment. But you would have thought they actually made life in the test tube based on the headlines. The proposed pathway for life. So how do the evolutionists explain this today? They say, well, you know what happened is that RNA catalyzed itself. So we just have to first evolve RNA. RNA then produces a protein. And then that protein produces, then it will make DNA. So it's an RNA first hypothesis. So you first have to come up with an RNA molecule. Now this is a very, very large molecule. And it has to be assembled. So you figure out how to get to this large RNA molecule. RNA is RNA for a reason. It is the most unstable of all the biological large molecules. If you are a chemist or biochemist in the laboratory, the last thing you really want to deal with is RNA. It is so difficult to handle because it degrades very, very easily in the laboratory. And there's a reason for it. It's unstable. The sugar backbone of it is a carbohydrate backbone. Remember I mentioned that. That carbohydrate backbone is very unstable, and it's used just to send messages. You want it to break down from the DNA to the protein factory, the ribosomes of the cell. So you just want it to be transitory. You don't want it to be long-lived. And so it is unstable on a long period of time. Whereas DNA it has a stability to it because it's a different carbohydrate backbone. That's the whole difference between RNA and DNA. DNA is called DNA because it's deoxyribonucleic acid. because it takes away one of the oxygens in the carbohydrate to make that ring stable. Just keep that in mind. So here, now scientists are saying, well, you know what, it was RNA that survived and catalyzed itself for all those years by itself until it finally, and this inside some kind of enclosed structure that kept it there. A micelle, what we would call a micelle, has to have some kind of membrane, a cell membrane, but there's no machinery to actually, because you need proteins to support the cell membrane. So you don't have a mechanism to support the cell membrane in which this is going to happen in the first place. So that was the whole idea. This is the concept of abiogenesis, is you have a reducing atmosphere, development of complex higher order molecules. I'm giving you the latest evolutionary explanation. Concentration of precursor inorganic chemicals in a primordial soup of pools, methane, ammonia, hydrogen sulfide. You have an energy source, lightning, heat, solar energy. Formation of simple, symmetrical, carbon-based molecules, formaldehyde, acetaldehyde, all of these are bad. Continued concentration of higher order organics without hydrolysis. Reduction, proteolysis, the reverse reaction. Yeah. So all of this is going uphill. So continued concentration of these higher order organics. So now that I've made a formaldehyde, now I've got to make an amino acid. Then I've got to make, I've got to make a carbohydrate. I've got to make, and you have to continue to grow these into very large, just the single units. Then you've got to bring those units together of hundreds of amino acids or hundreds of nucleic acids together in a large structure that can somehow impart information. And so then more recombination. So I'm going to end here. This is my this is my critique of abiogenesis. I haven't published it, but you're just going to have to bear with me. This was me being me, and so I'm going to. This is my critique, and I'm going to read it for you. Just a few little slides. So well, actually, I have this first, and then I'll read. Formation of chirality. This is a problem. Chirality is, you've heard of left-handed and right-handed, left-handed and right-handed molecules. And in life, all your amino acids are left-handed, right? All the but randomly making would be right-handed and left-handed. And so if you were just to randomly make two molecules together that makes something that's right-handed or left-handed, 50% would be right-handed, 50% would be left-handed. So how do you get all of life? All of life. Not just one molecule, but every single amino acid is left-handed. How does that happen? So this is a problem. Chiral catalysts. I'm not going to go through all these. Formation of simple amino acids because of lack of time. Even a carbon-containing acid attached to a reduced nitrogen. Formation of 50-50 racemic mixture. I was alluding to that. Formation of simple sugars. Even the formation of ribose, which is a very, very simple sugar, becomes very, very complex in a reducing atmosphere because this is highly oxygenated. Sugars are highly oxygenated, and you're trying to put this together in a non-oxygenated environment. Still continued concentration and sequestration of more complex molecules and the formation of what we call heterocyclic aromatics. There's just, it boggles the mind that we would even propose as scientists that we're going to try to do this randomly. And these are higher order molecules. So aromatics, sugars are hydrophilic, right? They're water loving, oil hating. So this is how do you get these sequestered in the right place. And aromatics are hydrophobic, so they're just the opposite. So how do you bring these together randomly? Protection from harmful UV light, formation of fatty acids, all of these things. So I'm going to, and I can just go on. Directed energy source, we already talked about that. Formation of polyribonucleotides to become their own enzyme is, catalyzing their own replication. the introduction of information. How do you even introduce the concept of how the information it would even know to form the right structure to catalyze itself? So self-selection being random-based changes have greater fidelity of replication but won't create variation. So that's another issue I could go on. So I'm going to put this in my summary. The 1957 life in a test tube experiments by Miller show only the formation of, it wasn't you, right?
My brother.
Sorry, I couldn't. All right. It's 1957, so I thought I think you'd be in grad school.
I thought it was earlier than that.
No, it was. So the 1957 life in a test tube experiments by Miller show only the formation of precursor organics related to simple amino acids, which supports the concept of early protein formation. Others have followed up on these experiments to varying degree of success, even obtaining some short polypeptides under tightly controlled, contrived, and assumed conditions. However, biochemical evolutionists have abandoned the protein-first concept of abiogenesis because of the failure of any reasonable mechanism to obtain a self-replicating beginning for proteins. So evolutionists favor now the self-replicating ribozyme hypothesis, which postulates a nucleic acid pathway for life's first sputter. This proposed pathway is actually a more difficult enigma for biochemical evolutionists to solve. At best, it posits the coevolution of complex, asymmetrical, and unstable sugars with high-energy phosphate bonds. I didn't even mention that. Hydrophobic, heterocyclic aromatic bases, and amorphous, short-lived lipid micelles, all without the myriads of reducing and oxidizing catalysts required to form the first nucleoside of the first ribozyme, let alone the whole ribozyme. So you might not understand all those words, but you know where I'm going with this. To compound the matter, the formation of the first working ribozyme from random combinations of a pool of available nucleotides introduces the problem of information. How does one get from the random combination of nucleosides to the first gasping ribozyme of hundreds of nucleotides long, all in a hostile and destructive environment? Remember entropy. Time doesn't help. The fact is, time is really the enemy, not the savior of the process. For as quickly as the high energy structures form, the reverse process is quicker still. This is the equilibrium problem, especially for the easily hydrolyzed ribonucleotides. Furthermore, even if magical catalysts were present to facilitate the reactions required to make the first polynucleotides, they can never, by fundamental understanding of chemistry, ever move the chemical equilibrium point. This cannot be understated. These reactions are all energetically unfavorable reactions, and any accumulation of higher order compounds would quickly revert to starting materials in the presence of any catalyst in time. Where does the direct energy come from to drive such reactions? Where is the ATP, or the metabolic processes and enzymes that form ATP? How will the ATP used without the enzyme? Even overlooking these obstacles, just about done. This is it. Even overlooking these obstacles, once a supposed self-interacting ribozyme has been reached, dubious self-selecting mechanisms to ensure survival of the best nucleotide combinations from billions of possible combinations, literally billions of possible combinations, for self-replication assume an insurmountable mountain of order, energy, time, and information just to get anywhere near a viable and survivable biomacromolecule, just one of these macromolecules from which to start the entire process. And all this sequestered in an eon-lasting, non-permeable prototype micelle membrane, unsupported by protein structural scaffolding. How does one seriously posit this as a mechanism? I suppose if one has already assumed methodological naturalism and Darwinism, he can make no other conjecture. He is trapped in a godless, myopic understanding of science. And they want to call me the flat earther. Right? That's how, this is where our science is today in the universities, because they've already assumed that evolution is the process. I go back to the beginning, what Francis said, we look at it, we see design, but the scientist must constantly remind himself that it is evolution and not design. And that's the bottom line. So I apologize that this was a little deep today, but that was my soapbox. So
So you
anyway,
eliminate
we're gonna-
God. You take God out of the picture.
yeah, that's what they want to take God out of the pocket. Well, and
Yes,
yes.
the more you understand the biochemistry and physiology of what you're talking about, just a simple cell and all those components that are in the cell, it takes an absolutely incredible amount of faith
It
to
does.
think that it happened on its own.
Yes, absolutely, absolutely.
Who's plant life?
Yes. Should you close for us, Charlie?
Sure. Lord, when we see this kind of information, what does it do to our minds to think how somebody could believe in anything other than God? Lord, you are so gracious to us in that you have created us so complex as we are. And we're thankful that you continue to providentially care for us with all the things that go to make life possible, for our food, for our atmosphere, for the energy that we have, for all the multitude of things that go together to make Earth possible, to make us livable. One little thing out of whack, and we wouldn't be here today. We thank you for your providence, we thank you for your care, we thank you for your grace, we thank you for the mercy that's shed upon this place today. Be with us as we go to worship, and thank you for all the preparation that went into this Sunday school for Dave's efforts. It's in your name we pray, amen.
Amen.
To close.